Hey guys, it's Miss Lewis, and today is our final day of teaching brand new content. For the rest of the year, you guys are going to be doing makeup work. If you don't have any makeup work, great, then you're done. But if you do, this is your chance to improve your grade for the marking period 4 and in turn your final grade. I could have given you today to do makeup work, but I wanted to squeak in as much content as possible because I know you guys were kind of shortchanged this year. So today we're going to be talking about RNA. RNA is something different than DNA. They're two different things, so please don't get them confused. Um, so basically, making proteins begins with copying part of the nucleotide sequence from the DNA to the RNA. So you're making copies essentially. And the main differences between RNA and DNA, other than their initials, is that the sugar in RNA is ribose as opposed to deoxyribose. So DNA is deoxyribose ribonucleic acid, and RNA is ribonucleic acid. So RNA is generally single-stranded. So as you saw in the previous lecture, it is a double helix. DNA is a double helix. So it's kind of two posts and like a stairway between them. RNA is just one post. It's a single helix. And it also contains uracil in place of thymine. So the nitrogen bases, guanine, adenine, cytosine, and thymine, it is instead uracil, adenine, cytosine, and guanine. So there is no thymine in RNA. RNA is a working copy of a single gene, and they can produce thousands of RNA molecules, a single gene. So RNA does have functions, and its main function is protein creation, creating proteins. And assembly of amino acids is also controlled by the RNA. So there are 3 different types of RNA. There's mRNA, rRNA, and tRNA, or messenger, ribosomal, and transfer, respectively. Messenger RNA is basically the messenger to from point A to point B. It's actually carrying copies of the instructions to the cells. So it's delivering a message. Ribosomal RNA makes up the majority of the ribosome, which are in cells. You guys should know what ribosomes are. And transfer RNA transfers amino acids to the ribosome. Don't get messenger RNA and transfer RNA confused. Transfer is actually transferring the amino acids, but messenger is just carrying a message. So there are 2 different processes that we're going to be learning about today, transcription and translation. Don't get them confused. Transcription is first, and we're going to talk about that now. Transcription is a process of copying part of the DNA sequence into a complementary sequence in an RNA. So it's taking the DNA and it's decoding it, essentially, the ATCG, and it is creating a sequence that goes with an RNA or can become an RNA. RNA polymerase binds to the DNA and it separates the DNA strands, so it kind of like unzips it essentially. And the RNA polymerase uses the DNA strand as a template. It kind of uses it to copy from. RNA polymerase only makes RNA copies at promoters, which is a specific base sequence. We're going to talk about that. So here is an actual depiction of the DNA, which is the double helix. You could see it has the blue sugar base and the red one. And transcription is basically turning this into this, a single-stranded messenger RNA. And as you could see, the thymine, the yellow, and the pink is now replaced with the purple on here. And this is just a transcription of the base pairs, the nitrogen bases. As you could see, the C is matched with the G, and instead of the A being matched with the T, the A is now matched with the U here. Okay. Now we have two things called introns and exons rather, when RNA editing is occurring. So introns are pieces of RNA molecules removed from the sequence. So they're things that are actually taken out. Think of if you've ever taken like video production or something and you have a video, you could take pieces of that video out. You could splice the remaining videos together to create one cohesive video. So the introns are the actual pieces that are removed, and the exons are the pieces that remain, and they make up the final messenger RNA. So these little yellow pieces are the ones that stay, and the blue pieces are the ones that are removed, and these yellow pieces then come together to create the final mRNA strand. The genetic code. So genes are just really codes, and then they are expressed through our phenome. So proteins made by joining amino acids into long chains called polypeptides. These properties of proteins depend on the order of the polypeptides. So the order can differ, and with each order of polypeptides, you get a different genetic code. So codons are the order of these polypeptides. There are 3 letters, and it contains, the U, the G, and the A and the C, and it specifies a single amino acid. So each codon is specifying an individual amino acid, and there are 20 different ones. Don't go crazy looking at this chart, because you don't have to memorize this, definitely not for this year because this year's over, but you have the U, the C, the A, and the G, uracil, cytosine, adenine, guanine, and you have it going down the side as well. And it's kind of like a Punnett square, kind of like a giant Punnett square, and you cross these over with these, and you come up with all of these different 3-letter combinations. This is just another way of looking at it. It's the same thing, it's just a different way of presenting the information. So for example, this is just an example, a sequence of bases in RNA, UCGCACGGU, is read as UCG, so you separate the first 3 letters, and that's serine. CAC, the next 3 letters, is histidine, and GGU is glycine, and those are the last 3 letters. So you're separating every 3 letters. AUG codes for methionine, but is also the start codon. So there are starts and stops. The RNA genetic coding and the transcription can only begin on certain amino acids and end on certain amino acids. So this is the one that it can begin on, the transcription, and these are the ones that they can end on, the UAA, UAG, and UGA. Translation. This is the second process that happens with RNA. So the nucleotides in mRNA serve as instructions. We talked about how mRNA are actually delivering the directions. So translation is the decoding of those directions of mRNA into polypeptide chains or proteins. And this takes place on the ribosomes that are in the cell. Translation uses information from the mRNA to produce proteins. So it actually takes these directions and produces proteins. And there are a few steps in doing this. So, number one, before translation, mRNA transcribed in DNA and released into the cytoplasm. So, the mRNA is set out from the nucleus where the DNA is and is released into the jelly cytoplasm within the cell. Number two, mRNA attaches to the floating ribosomes that are, floating around the cytoplasm. As codons move through the ribosome, proper amino acids enter the ribosome to attach to polypeptide chains. Transfer RNA, so the tRNA, which we haven't really talked about yet, puts the correct codon onto the RNA strand. Anticodons are three base pairs on tRNA complementary to the mRNA codons. So they're kind of like the opposite of the base pairs, okay? So like the U would go with the A and the G would go with the C. And number three, ribosomes form peptide bonds between amino acids. Also, the bond holding the first tRNA to its amino acid is then broken off. And number four, the final process is polypeptide bonds continue to grow until a stop codon on the mRNA is reached. So the stop codon is one of these three, the UAA, the UAG, and the UGA. So it doesn't stop translating until one of those codons is reached. So DNA is known as like a master copy, and RNA are just the blueprints of that master copy. So many proteins are enzymes which catalyze and regulate chemical reactions. I believe, okay, this is our last slide, and this is just a depiction of it. I will be posting a video that actually shows like an artistic rendering of what happens because it's kind of confusing to read. It's much easier to see it happening, see the RNA kind of moving through the transcription and translation processes. It's just an easier way to understand it. So I am going to post that and I want you guys to watch that as well. Okay, if you have any questions at all, please let me know. I am still available for virtual office hours if you guys are interested in that. Also, I'll be reaching out to you guys individually. If you have any missing assignments, I will make you aware of those. Okay, so again, if you have any questions, please let me know. I look forward to seeing you guys when we get back to school. I will, make sure you come and stop by and say hi. I know I wasn't with you for that long, but definitely do that. I hope you guys have an awesome summer and stay safe.